Huntington disease is a monogenic disorder that follows an autosomal dominant inheritance pattern, meaning only one copy of the defective gene is required to develop the disease. Prevalence is approximately 1 in 10,000, with offspring having a 50% chance of inheriting the disorder from their parents. Huntington disease is a neurological disorder that causes the progressive neurodegeneration of the central nervous system. Disease onset is usually between 35 and 55 years of age and progresses unstoppably for an average of 15 to 16 years, resulting in fatality. Those affected with Huntington disease do not die from the disease itself, but from associated symptoms and health conditions, including dysphagia, heart failure, infection, and in some cases, suicide. Technological advances of gene mapping in 1990 allowed studies to isolate a large gene on the short arm of chromosome 4. Using cloning and sequencing of critical regions, a mutation now identified as the Huntington gene was located, consisting of an expansion of trinucleotide sequence CAG in the 5' region, encoding for the amino acid glutamine. This mutation leads to the toxic gain of function of the Huntington protein. The amount of CAG repeats expressed has a direct relationship with age and severity of onset. A rare onset of Huntington disease between the age of 8 and 21 is described as juvenile Huntington disease and is associated with 50 or more repeats of the CAG expansion. Symptoms of Huntington disease are caused by neuronal death in the basal ganglia, cerebral cortex, and other regions of the brain. The cognitive disorder gradually progresses with loss of neuroplasticity and slowing of intellectual processes, eventually advancing to severe dementia. Symptoms include involuntary movements of the body, and steady neurodegeneration that leads to behavioural and cognitive impairments. In later stages of Huntington disease, neurological function is severely impaired, which leads to changes in comprehension, memory, judgment, and reasoning. Individuals will eventually lose their ability to walk and talk, have poor dietary intake due to dysphagia, as well as loss of independency, which results in requiring long-term assisted care. The area of the brain most affected by Huntington disease is the basal ganglia, a group of nerve cells at the base of the brain that coordinate motor function within the body. In advanced cases of Huntington disease, the brain weighs 25 to 30% less than a healthy, non-diseased brain. Currently, there is no cure for Huntington disease, leaving those who may have inherited the disease in a challenging position whether to undergo testing. Pre-symptomatic genetic testing is available for people who wish to know if they have inherited the mutated Huntington gene. Testing will indicate the number of CAG repeats in an expanded Huntington gene and thus give an indication of whether a person has the condition, age of onset, and an approximate severity. Genetic testing allows those at risk of developing symptoms of Huntington disease to make informed decisions about their future, such as medical, financial, and legal decisions. Pre-implantation genetic diagnosis has the potential to prevent passing on the mutated gene to children through the use of in vitro fertilization to ensure the fertilized egg does not contain the mutated Huntington gene. Prenatal genetic testing may be performed to detect the Huntington disease mutation by chorionic villus sampling or amniocentesis, which involves testing a sample of cells from the chorion or amniotic fluid. Current research surrounding Huntington disease is focused on treating motor, cognitive, and psychiatric symptoms, along with therapies aimed to modify the disease at a molecular and cellular level using genetic and non-genetic approaches. These treatments and therapies aim to lessen the severity of symptoms, slow the progression, and provide a better quality of life. The expression and regulation of microRNA is uncontrolled in the Huntington gene. Due to large quantities of microRNA being found in the hippocampus and cerebral cortex, it is indicative that microRNA plays a role in memory, learning, and motor function, making it an area of interest. Along with microRNA, small interfering RNA assists in gene silencing, gene regulation, and synthesis of proteins responsible for maintaining synaptic plasticity. Scientists have had successful trials in Huntington disease animal models using RNA interference. They use microRNA and small interfering RNA molecules as biomarkers to target the breakdown of messenger RNA prior to translation by activating the RNA silencing complex. Improved motor function was observed in an animal study using Huntington gene silencing, and the mutant Huntington protein was decreased by 50%. There is still much to learn about Huntington disease and its molecular mechanisms, and researchers are hoping to discover an effective treatment to alleviate symptoms, slow disease progression, and ultimately find a cure, giving individuals and their families affected by Huntington disease a better quality of life.